Okay, so good afternoon, everyone. My name is Ursula. I am the assistant in the OIT Boost project, that is the organizer of today's event, and I'm happy to welcome you to the webinar in the topic of openness and transparency agreement on animal research and science that will be presented by our wonderful speaker, Dr. Marcello Raspa from the University of Milan. Before we will dig into the topic and to our speaker as well, just a couple of words about the OIT Boost initiative. This is the 3 years project funded from the Horizon Europe program that aims to develop research and innovation excellence and improve the management capacities of Estonian University of Life Sciences and University of Tartu in the field of One Health. And this is happening through the partnership and collaboration with top experts in this topic, which are the University of Helsinki and the University of Milan, whose representative we are hosting today. So I'm happy to welcome you once more to the webinar and present our speaker, Dr. Marcello Raspa, who is Executive Technologist at the National Research Council, Institute of Biochemistry and Cell Biology. Dr. Raspa is also expert in research ethics and dedicated advocate for the ethical and transparent use of animals in science, actively contributing to the national and European Union level committees on research integrity and scientific ethics. And as I heard from the speaker before starting the webinar, he would like not to only present you some information and data, but also would like to discuss and talk with you on the topics that will be presented. So anytime you would like to provide a comment or have a question, please use the button React in Zoom opportunities or write your question or comment in the chat. Besides, I wish you interesting and fruitful discussion, webinar, new knowledge. And now, Dr. Raspa, the floor is yours. Please go ahead.
Thank you, Ursula. Very nice introduction. And so can you hear me well? It's okay?
Yes, it's very nice.
Okay, thank you. So let me thank the University of Milano and even the Professor Tiziana Brevini that invited me to this talk. So today I would like to talk about the recent achievement in the Italian scenario. And this process is commonly known as Openness and Transparency Agreement in Animal Research and Science. And this because the effective communication and transparency are crucial topic in fostering public understanding and obviously the trust in science, in animal research, welfare and care. So it's very important, this process that has been achieved since a few years, even because the scientists were always in the tower in the past. Now are trying to communicate with politics, with the media, with the youngers especially, but even with other stakeholders. And the challenge is to highlight the ethical practices to researchers that can build dialogue with the stakeholders as well. Okay, so in this talk, we will have a short overview of a transparency agreement, communication initiatives. Then I would like to get through the achievements with the technologies, methods, and the recent process in Italy. I would like to continue with showing recent achievements that I got at the University of Bicocca in Milan about the preclinical infrastructures, international infrastructures of research, and the conclusions will go on. As Ursula pointed out, I will leave you all the materials, all the slides, and whatever you want. I'm a public employee, a public officer, since I'm a technologist at the National Cancer Research, and this is part of my duties, so communication and even the interface with the public, with students in different kind of projects. And I like to discuss, I'd like to talk with people instead of reading slides for hours. This is my intention. So if you want to interrupt me, it will be a great pleasure for me to answer if I can. So you know that science is open, and this is the most powerful antidote to anti-science. If scientists don't work, don't talk, the anti-science will go on. And this is the prerequisite for that the transparency could declare. And in science, the transparency encompassing the open sharing of data, methods and results, even negative results. And this is very crucial for fostering the trust with society and politics first, reproducibility, and ultimately the advancement of knowledge. And other researchers can even judge and try benefits on the discoveries and experiments that have been done by other scientists in the same community. So the Italian, for people that don't know my institution, the Italian National Research Council is the largest multidisciplinary public research and technological development institution in Italy, has been founded more than 100 years ago and plays a vital role in driving scientific advancement and fostering technological innovation, especially in different kind of areas like material science, nanotechnology, biotechnology, and promote collaborates with the other international scientific with exchange and promoting research projects. CNR has about 8,000 employees and is divided in 8 departments in the whole country. So this is the real news. The health ministry in Italy has been finally agree on publishing and promote a concordate on transparency this year, so the 2025, for the scientific institutions that use animals in science, animal models in science. In this agreement, the ministry invite other centers of research to commit and communicate clearly the use of animals with the society, with the politics, with the media. For what concerns the agreement, the countries with the transparency agreements in Europe are the following, the pattern. So the first was the UK in 2014, and last year there was a big celebration of 10 years of transparency. Then we'll join Spain, Portugal, and Belgium, and then the other countries that you can see in this slide, including 2 extra EU countries very far away, New Zealand, Australia, and finally this year, Italy. More countries will come, Ireland, USA, Canada, Austria, Sweden, and Greece in the following years. And I can say that more than 500 institutions have signed a TA in the EU. This slides, this material has been provided by EARA, the European Animal Research Associations, led by Dr. Kirk Leach, and this is one of the achievements that they make through a survey last year. They individuate in different countries the lack of communications, especially for statements on the institutional websites that has been assessed in 2023. You can see in the slide that, for example, in Italy, less than 20% of institutions that use animals don't have statements on their web page. This is the basis, including the CNR, of course. So this is very nasty, it's not nice, and it can be criticized by the community, especially the community that provides support, financial support to us and to the world institutions. So EARA is probably the most important stakeholders, the most important association active in this field. And EARA lead and trust the voice in Europe representing organizations that use and breed animals for scientific purposes, drive the efforts to respond to the growing political pressure and campaigns that premature seeking and the use of animals for scientific purpose that is unrealistic and drive cultural change, focusing on institutional policies and practices on how organizations communicate to the public about the use of animals for scientific purposes. And at the end, this is important as well, support and lead efforts to maintain a conducive operating environment to the access and use of animals in scientific research in Europe. So toward the openness and transparency, we need the way how to get there. And this is crucially important at the same time. What's about the transparency agreement? This is just an example that has been this coming from the Netherlands. And the agreement is a sort of contract, not legally different legally requirements, but obviously is different in different countries, especially in Europe, and is a voluntary agreement with no legal, strict legal requirements, and everyone progresses at their own pace according to the possibilities and limitations. And the signature can be made at the university, public research institutes, industry, so companies, the supply chain, and even the funding bodies, medical charities and patients groups. For what concerns the agreement, all the countries that are where the agreement has been established agree on 4 commitments. First, to be clear about when, how, and why the animals are used in research, enhance the communications, especially the professional communications, not very common in the scientific environment and among scientists, with the media and the public about the research using animals. So all the signatories will be proactive in providing opportunities for the public to find out about research using animals. And finally, everyone commit to report progress annually and share the experiences. Beyond these commitments, it will be it has been established a network that represents each signatory institute with researchers, animal welfare experts, and communication experts within institutes and between institutes. And this is, for example, there is a transparency agreement meetings regularly established by EARA, for example, and even the Congress that will be in this year, at the end of this year, in November, in Germany, if I remind well. This is the example, some practical examples. This is a group of science and society in my institute, the Institute of Biochemistry and Biocell in Rome, at CNR. And we established a voluntary group of scientists that public outreach and communicate with the social media. At the bookshop events like the Fiera del Libro in Roma in 2024, the European Research Night in 2024, and of course the Openness in Animal Research that has been realized in 2025. Even they organize events at the high school, students and teachers with different kind of format and events communicated through social media and even are active in editorial contribution in books and in reading and editing some paperwork. For what concerns the events with the schools and the public outreach events, you can see in the slides some of these events that has been organized in the city, in Rome, but even through media, to the media channels, and even abroad. And this is very just an example, but can be organized locally, but can be can be promoted, can be analyzed in another context. So about the project that I'm part of, because I'm the CNR, I work for the European Mass Mutant Archive and Infrafrontier, that is a repository of mouse, of rodent genetic resources for researchers. And we have a vast collection of mouse strains, each carrying a specific genetic mutation for research applications. They are invaluable models for studying biological process, mechanisms, and treatments with a vast genetic diversity. And we offer even access and support to researchers through researchers, and we contribute to the advancement of biomedical knowledge by this project that is active since 2001. For what concerns Infrafrontier, EMMA is part of Infrafrontier, and that is called Archive Frontier. Our models, as you can see in the slide, we are part of different countries. At the beginning, we were 7 institutions. Now we have 22 partners in 15 countries, in 2 continents, and this big community that are linked to the mutant genetic resources, mouse mutant genetic resources, are spread in different kind of countries. And even part of the activities of communicating activities and openness and diffusion of information are part of this project of transparency. For what concerns the communication policy, first of all, we have an international group, the Animal Welfare Expert Group and Technical Working Group, and we work studying, defending, and verifying principles and procedures in different areas, phenotyping, cryopreservation, etc., with different kind of trials considerations. Recently, we organized a session at the FELASA 2025 and present 4 talks on the topic, on trials topic, covering scientific support offered by Infrafrontier. And we support and join the EU with initiatives, especially even in conjunction with the EARA, when the European cities initiatives on animal research phase out that got strength and was contrast even from by the colleagues at the EARA the years before. We coordinating with think with the EARA research centers and public institutes, participate every year in the board Be Open Animal Research Day campaign moved by ARE, posting about trials and animal welfare in Infrafrontier network. For what concerns other topics, as you can see, we work even in the alternatives. One of our members was a winner of the 2020 ICT Trials Prize. There are a lot of informations concerning the trials and alternatives to the animal use and ethics in our webpage, like this one. And we are very active, very promoting, and very positive, I can say, in finding the way on how to reduce the animal use and how to refine the procedures, how to avoid the use of animals in procedures. So this is the state of art concerning even some examples of my work, let's say. So Infrafrontier and the CNR and my institute. For what concerns the one survey that I made in conjunction with Bicocca University last year, I interviewed some participants, some stakeholders, with and the data were analyzed by using a qualitative analysis supported by colleagues. And I interviewed 20 research infrastructures, opinion leaders, stakeholders from 13 countries in 3 continents, accompanying with the literature analysis and researches on the internet, web, and databases. And we conclude that the most suitable model for, the research infrastructures for the future sustainable development is in line with the ethical principles of 3Rs and green economy. And this is for the future. So the research infrastructures that is one of the most powerful tool in science, especially in Europe. They offer support and methodologies, personnel, equipments for laboratories that cannot have this kind of tools by and the work even by implementing, investing in procedures and services with strong application of technological development and innovation, especially with artificial intelligence and automation. We invested, I discovered even that these 20 opinion leaders found very useful to invest in the communication collaborative plan. That is the main prerequisite for recognition and thus for gaining national international reputation and credibility. So the new infrastructures, the modern infrastructures of research will take care of ethics and 3Rs and policy with the eco-sustainability and ecology and with the relevance of technological impact and knowledge development. I can steal you a few minutes more to give you some practical examples on working in 3Rs in the group, in our group of research, technological research. So we are very involved in automated home cage monitoring since in the recent years there has been a growing consensus that in order to improve the accuracy, robustness, and reproducibility of results, animals should be left undisturbed as much as possible and not impact their state of stress, monitored 24 hours per day, 7 days per week, and particularly at night when they express their behavior at most. Take into account that historically scientists take care of animals few hours per week and only in the day, not in the night. And rodents are night species, night animals. For what concerns the reproducibility, the housing matter is the hidden factor that's shaping the reproducibility in animal research. It's very important to analyze animals in their home cage environment. And is significantly influenced by housing condition at a factor that often overlooked in experimental design. By controlled variation enhancing animal welfare, it's possible to make studies more accurate, more precise, and more robust. In terms of reproducibility, and for what concerns the translation medicine, is a fact that a lot of people has been published that preclinical tests fail for the 85% of studies. This is dramatic. Not helps our field. This is not versus only the scientific community, the patients and the companies, but even in terms of wasting of resources, fundings, and workload. This is one the fact that we have to take care for the future. This is called a valley of death in preclinical research. It can be improved, especially in order to improve and make more significant the clinical trials, and we need to improve study design. For what concerns our work, so that's because we insist in home cage monitoring and in the welfare of animals. This is another example. We recently studied the vocal communication in mice. We use very new tools, very modern tools, even because the vocal communications plays a crucial role in parental care and maternal behavior, even because the neonatal mice are capable of producing vocalization in a wide range of frequencies. So we use the artificial intelligence, and therefore through the convolutional network, neuronal network, we established through a spectrogram and noise filter the way on how to intercept in the cage the vocalization of neonates and that give birth. And we are now able to identify to perform in real time the birth of mice through these graphs, for example. And the parturition, we make the study multicentric in different countries, different centers. And now we validate this system that is very important next to the other procedures with a more different kind of scientific group of scientists. Another practical sample is this one. The beginning, we use a lot of mice to bank, to archive one strain. We use basically from 60 to 70 mice in 6-8 weeks, in 2 months, basically, with 5-10 males, so plus 50 females. Today, in 2 months of work. Now, after the 2008, 2010, with new methodology that we developed, now we are able to cryopreserve one mouse line by using only two males and 5 females, but through the IVF, in vitro fertilization. That these results seems to be a miracle, but has been achieved with a long run of work made in different kind of countries and different kind of groups in our network. These are practical examples that show how to reduce the number of errors that work in the welfare of animals as well. Even the shipment of mutants, and now most of the times they perform through frozen stocks, embryos and sperm, in worldwide, of course. And next to it, we work in different kind of fields. These are some examples, but we publish a lot of papers only by refining technologies and methods and try to reduce as much as we can the number of animals, financial support, so the effort of work, of staff, and from different kind point of view. In conclusion, and for the future, we can state that the study demonstrated the feasibility of using 3 R's approach and alternative methods that can lead to wider replacement, reduction, and refinement is possible. Not the phase out of animals is possible. This is realistic now. It's impossible to mimic the complex of one animal, one chip, one organoid. But it's possible to reduce sensibly the number of animals used in science. We need better communication strategy, especially in scientific world, the scientific staff. Scientists can communicate, even not too much with other scientists, but not very well with society, with the youngers, with different categories, workers. We need more openness and transparency. So this process can be crucial for the future, for building up trust and even more support from politics and from the society. Even the continued collaboration between research institutions is crucial for advancing technology and animal welfare. So I just give you some examples of this work that has been made in collaboration with the different kind of centers, and so this work always it's very powerful and valuable. Thank you very much. So I try to be as fast as I can with the time. I hope to be in time, and so open for questions. Thank you.
Thank you very much, Marcello. So, do we have any questions, any comments from the audience? Anyone who would like to share something, add something?
Can I just make a comment? Well, just an
Yes.
observation in general, if you don't mind.
Yes, of course, Tiziana.
Marcello, how easy do you think is to be able to standardize, let's say, all the different aspects when you are trying to apply those to such a wide number of institutions with very distinct and specific reality?
Thank you, Tiziana. This is a very interesting question. So we work on it several times. Sometimes studies failed. For example, we make some trials. You know the Jackson Laboratory, the largest institutions in the world, probably with mutant mice. So we make studies at the Jackson Laboratory, the same studies, same materials, same animals, same conditions. JAX, MRC in London, and CNR in Rome. And all of three, we got different, completely different kind of results. This was made, unfortunately, by the environment, caused by the environment, because the Jackson Laboratory, where my education come from there, and the Jackson has some rooms with hundreds, thousands, millions of animals. With a lot of traffic, a lot of operators. At the SNR, vice versa, our breeding rooms are very quiet with few animals, few personnel that is allowed to enter. Sometimes this collaboration was fruitful. For example, we established a way on how to ship, instead of shipping live colonies, to shipping frozen materials in dry ice, -80, -79, and we make these trials in 5 different centers in the world. This was very successful. But taking into account that I've read some interesting findings, for example, that a lot of experiments that that have been realized in different laboratories cannot be replaced. It cannot be repeated in other laboratories for a large percentage. That means that conditions sometimes are different.
Thank
Yeah, I
you.
think this is a very different, this is obviously a very important aspect, especially if you consider that most of the listener here comes from laboratories scattered around Europe. So you would expect significantly different conditions. And I don't know. I think, at least in my experience, even when you try to transfer a protocol from one laboratory to the other one, you tend to need adaptations and slight changes that then tend to become more and more along the progress. So yeah, I think that will take a lot of effort. That was my point.
If I can add something, is actually our procedures are well organized and proved, and we used to validate these procedures in different kind of laboratories usually. Only things that are working in different kind of environment and are effective have been published.
Yeah.
This make very helpful credibility and trust in our work, and things are working very well in this way. It's not always possible in science because it takes money, efforts, stuff, etc. But I think that this is the way on how we can go. We have to go in the future because there is not too much credibility and trust on experiments, especially in some parts of the world, unfortunately.
Mm.
This is my opinion, personal opinion, but I made a lot of studies in multicentric way.
Can I
Yes,
Yeah.
please go ahead.
follow in the same line of thoughts and taking advantage of Marcello experience to also ask him another aspect that we've been involved with directly. So we are developing in vitro models with the clear aim to reduce the animal studies and the animal experiments, the number of animals which are used for in vivo trials. And we face a number of problems, among which is that even in vivo studies give different results depending on the parameter you're looking at. So therefore, once you want to compare an in vitro study and an in vivo studies, since the data that you take out from an in vitro model are very different from the data you take out from a whole organism, from a whole animal, so you cannot compare directly. And then it comes up the interpretation, which aspect is more relevant to the others. And so the problem is, okay, the criticism was, oh, your model is unable to predict the results that you would have achieved in vivo, but which is partially true, but it's also partially true that we didn't have a single in vivo outcome, a multiple in vivo outcomes. So some of them we got right, some of them we didn't. So the problem is, how do you measure the efficiency and how do you compare two completely different sets of data? Is there any work specifically devoted to improve this aspect, as far as you can tell, or do you think it should be?
No, you catch exactly the point. So, for example, we collect in Infrafrontier EMMA, the European Mutant Archive, we have now roughly 9,000 models, no, in our liquid nitrogen tank. Now there is a new project that compare some procedures in live mice and in vitro. This is a project funded by the EU, and if some procedures are working, are exactly compared, animals will be avoided for the future. The project is called Prime Tech, and exactly the scope is exactly what you described right now. So this is what you pointed out is very important. It's very modern. It's very realistic. Unfortunately, in vitro, the system in vitro are not so refined and complicated as the life has.
Yeah, that's definitely an important point, but that is all. Well, our approach was always be to provide supportive data in order to reduce one of the R, reduce the number of animals to be used. And that again, I found that it's not so clear how to do that, because again, the set of results are completely different. So the expected differences, which is one of we want to do, is how to move from a difference in vitro to an expected difference in vivo, and you can use that in order to calculate the numbers of animals, or if you can reduce the number of animals, or making a learned prediction.
Yeah, take into account one simple example. Has been estimated that, for example, in the mutant world, in the mutant with the mutant mice, besides the numbers, from 50 to 75% of animals generated are not used in procedures for different reasons, different sex, or, for example, different genetics, different age, and this can be work on it. So it's a large numbers. For example, if you use 50 animals in procedures, in experiments, we have to generate 100, 150 more. So we this is this is a problem. It's a very key point to be worked with. And there are many aspects in toxicology, in pharmacology. There are many fields that we can use the reduction of animals and the final procedures, but not in the basic research. It's impossible right now.
Thank you, Fulvio. Thank you, Marcello. Do you have any other questions or comments or some experiences to share? I don't see, I don't hear.
Can I just add just an observation to add complexity to the already complex situation?
Yes, of course.
And I was just thinking that I mean, in the recent years, the models have become more and more complex, and of course, the use of organoids and 3D-dimensional scaffolds and all sorts of the so-called highly predictive models recently generated. Okay, I think that increasing complexity in these models has also led to increasing complexity in a number of accompanying techniques. For instance, one difficulty that we encounter is how to correctly cryopreserve these models. And I think this is a key aspect because many laboratories are now creating super complex, highly predictive models.
Complex.
That then are very difficult are very complex to cryopreserve in a way because you only know well and in a reproducible way the very standard traditional techniques, but then all of a sudden most of the lab tend to face complex structure, tissue-like structures that you want to store and to use, I don't know, in 1 year time or something. And yeah, this is also something I think we have to think about because I'm not aware of anyone really in control in this perspective.
Yeah, I agree completely. It's my life in the last 30 years. So this is, Tiziana, you point out a very crucial point because, for example, multiple mutants are very difficult to cryopreserve sometimes, even impossible in some ways. But for example, we are working on the different kind of paradigm now to cryopreserve strains. One of the major things is the availability of strains because the liquid nitrogen technology is very old, and we are working on getting the possibility realistic to maintain strains in the fridge. Amanus 80, for example, this can be realistic so far. We are studying it since 16 years, but it's very complicated. For example, since the complexity of mutants, especially humanized models, to cryopreserve in the good way, especially with multiple mutants. So we need not 1 hour of discussion, but 1 day or a couple of days. But you know that I know that you are expert on embryology and or the reproductive field, and so you know this better than me probably. You are right anyway, completely right.
Great, thank you very much.
Thank you, Ursula.
Maybe there is still any other comment or some new thought came to somebody's mind. If not, then thank you, Marcello, for sharing with
Pleasure.
us your knowledge and experience and perspectives. And thanks to all the participants for joining us today and for participation. As it was mentioned, the materials, the presentation from this webinar will be shared with everyone, and also recording will be added to our Onehealth Estonia YouTube channel, and link will be sent to you in a couple of days after the webinar. So thank you very much for today's meeting. I wish you nice afternoon, sun, and to, I think, many of you, nice holidays, nice summer, and see you on the next activities from OH Boost project. Thank you very much.
Thank you.
Thank
Bye
you.
bye. Thank you,
Bye.
Bye.
Marcello.
Thank
Bye bye.
you.